Okay. People are. Good morning. We will be starting the webinar shortly. Good morning, good afternoon, and good evening, and welcome to this webinar today. I'm Wafaa El-Sadr. I'm the Global Director for ICAP at Columbia University, and it gives me great pleasure to welcome you to a webinar today that's entitled Phylogeny and COVID-19, Shedding Light on Origin and Transmission of SARS-CoV-2. It is our great pleasure at ICAP to also co-sponsor this webinar with the Treatment Action Group, TAG, as well as also in collaboration with the COVID-19 Working Group in New York. During the webinar, please feel free to enter your questions in the Q&A, and we will have a chance at the end, after the speakers finish their presentations, to engage in discussion with both presenters. So please do enter your questions in the Q&A as we move along. I'd like to now introduce James Clerstein, who will introduce our speakers today. And James is the co-founder of Prep for All Collaborative, as well as also the co-founder of the COVID-19 Working Group. Welcome, James.
Thank you, Wafaa. Thank you, Wafaa. So we have we have two real sort of leaders in the field of phylogenetics and who have been working really on some of the cutting edge work that's being done in COVID-19. So our first speaker today is Dr. Bette Korber, who is a laboratory fellow at the Los Alamos National Laboratory in New Mexico in the T6 division. That's the Theoretical Biology and Biophysics group. Her work focuses on viral evolution, the human immune response to infection, and vaccine design. She leads an interdisciplinary team that provides bioinformatics, theoretical and statistical support in collaborative efforts with experimental researchers working primarily on HIV, but also on filoviruses, hepatitis C, and influenza. And then the next speaker will be Joel Wertheim. Joel is an associate professor of medicine at the University of California, San Diego, where he studies the evolution and molecular epidemiology of RNA viruses. His research primarily focuses on how molecular epidemiology can be used to inform HIV public health surveillance and prevention activities. In this capacity, he serves as a consultant to the CDC on HIV molecular epidemiology. Prior to the pandemic, his only research on coronaviruses was from the Cretaceous period, but he is now applying this approach to understanding SARS-CoV-2 molecular epidemiology and evolution. Dr. Korber?
Great, thank you very much. I'm going to try to share my screen here. Can you guys see it? Can you see my screen?
Yes, yes. I can see your screen. If you can just put it in slideshow, Betty.
Yeah. Okay. So what I was asked to speak about today is the phylogeny of the origins of SARS-CoV-2, and it's the virus that causes COVID-19, where it came from, what we know about it, and also to give you guys a little bit of background material before I plunge into that and talk a little bit about what a phylogeny is. So that's where I'm going to start. And what is a phylogenetic tree? And what is a phylogeny? Well, it comes from the ancient Greek, of course, and that phylog means a lineage, and the genesis part means origin. So it's trying to trace the origin of lineages. And I think Darwin gets the credit for the first phylogenetic tree. He wrote in his notes, I think, and then he drew one. And what this shows is sort of the foundation of everything we've built on since. And if you look at these little endpoints, they're called leaves or taxa. So they might be a group of related organisms if they're close together. And then as you get further and further away in the tree, you get to more and more distant organisms. So he might have been looking at something like the size of a bird beak and the shape of a bird claw and the color of a bird feather. We're looking in our cases at DNA sequences. And so I've got a very simple but very informative phylogenetic tree that has to do with SARS-CoV-1, the original SARS outbreak, on the right to try to walk you through how you might think about one of these constructions. So just like when Darwin had these close related taxa, we have very closely related SARS-CoV-1. So these would all be from humans from the first outbreak in 2002, 2003. They have a common ancestral sequence here. And these were consensus from different regions in China. These sequences were, so they're sort of representative sequences of different regions in the epidemic. I'm going to show you a larger tree of this in a moment, but you can see these are all very closely related. They both come from one ancestral sequence. And the way you know they're closely related is because of these horizontal branch links. So these horizontal branch links are estimates of the genetic distance. So this may have just had a single mutation from the ancestor, and this might have had a couple. And this sequence is more closely related to this ancestral strain along with this one. The vertical arrangement isn't so important. You could swap this whole thing out and put this one to the top, and it would still be the same tree. What's important here are these branch links. So these human sequences are all coming from the epidemic, and they were very closely related to a palm civet from a market. And so this was isolated the same year. And this sequence was the most closely related among the different coronaviruses, beta coronaviruses people were looking at. So they were able to determine that this human outbreak came from palm civet. And then if you went to the next year, there was again another much smaller outbreak. And you can see here is a human sequence from the 2003-2004 outbreak. And here's a palm civet from that same region. You can see that this human sequence is a lot closer to the palm civet from the same time. So this one did not evolve from the first human epidemic, rather was another crossover event. So you can learn a lot from looking at these trees. And these branch links have a scale bar down here that gives you a sense of the genetic distance and the number of mutations between these ancestral events and the taxa or the leaves. And so that's just kind of a rough sense of how to think about a phylogenetic tree. And you know, this is a different style of phylogenetic tree, and it comes from a wonderful review that was written in 2019. And we've had multiple crossover events from coronaviruses into the human population. And several of them are coming from alpha coronaviruses, and they just cause colds in humans, and those are marked. But the ones that have been really, really lethal, and the one that's causing the current pandemic are Cervical viruses, and that means they're SARS-like, and they're all of these beta coronavirus genera. And so this pink is the beta coronavirus. There are several common colds in this genre. There's the MERS. Merbeco viruses are the bat-related viruses, and the MERS virus that caused the MERS epidemic. And Cervical are the ones that caused the first SARS epidemic and the current one. And so what I'm going to mostly talk about over the next slides is how we got the current pandemic strain from this basic lineage. And as it turns out, bats are the main mammalian host of these things, the reservoir. They don't get sick with these coronaviruses. Worldwide, they carry very diverse coronaviruses. And so there's this incredibly diverse population in bats. And for the case of the first SARS epidemic in 2002, we only had slightly over 8,000 cases with a 9.5% fatality. And it seemed to have gone from this bat reservoir into civets and then into human. And this could be traced really directly to markets and even restaurants where these were served as food. The MERS comes from dromedary camels, so it's actually ongoing. It's very pretty rare. There's 2,260 cases as of last year with a very high fatality rate. And so these pass from camels to us. Okay, so here's that little tree again that I showed you. And this is from Song et al. Paper. And this is a tracing again of the first SARS epidemic. And so it went from bat into palm civets. And this shows you a phylogenetic tree of the different outbreak sequences during that first epidemic of SARS that we had in 2002 and 2003. So you can see the evolution in humans here, phase 1, phase 2, phase 3, and then you can see this little tree that I showed you before. We know that it came from palm civet in both years and was probably introduced through farming and food. So the phylogenetic trees are a good indicator of phase changes, but recombination is an extremely important part of the history of these viruses. And if you're not used to thinking about recombination, I wanted to try to give you a simple enough example where you could see it. And it's actually going on in our current COVID-19 pandemic. So I just took an example from COVID-19 that shows recombination. And the way you can think about that is if you have two viruses and they get in the same host, they can recombine so you can have part of one and part of another and get a new virus that's carrying some of the diversity of one and some of the diversity of the other. And that's a recombined virus. So if you look at the top, this red line is one virus. The blue line is another virus. These are from the Netherlands. So I got all these sequences from GISAID, and I'm very appreciative of all of the people who donate their sequences to GISAID worldwide so we can all study them. But what this is 30,000 bases, and every time you have a tick mark in the sequence below at the bottom, it's a place where the red and the blue differ. So these viruses don't have a lot of variation in them that are circulating. Out of the 30,000 bases, every little tick mark you see are the only differences between the red and blue. But this recombinant strain has all the blue-like bases on the right-hand side and all the red-like bases on the left-hand side. So if you look at a phylogenetic tree, it'll kind of confound the tree. So I've marked the recombinant sequences in this bright magenta color. And if you just do a tree of all 30,000 bases, there's this sort of red-like clade circulating in the Netherlands. There's a blue-like clade circulating in the Netherlands. And the clade just means a branch point in the tree. So it's a branch of related sequences. And a recombinant comes up halfway between, as you might expect, because it's carrying half the red-like form and half the blue-like form. If you were just to make a phylogenetic tree of the first 20,000 bases, our recombinant looks exactly like the red-like form. If you make a recombinant, a tree of just the last 10,000 bases, it looks just like the blue form. So you get a very different tree in terms of this sequence. It shows up in different places depending on where the recombination breakpoint is. So I just wanted to give this as a simple example. That's how recombination works. So the viruses have to get in the same host, or it might be a laboratory contamination that gives rise to something like this. But anyway, the viruses have to be able to recombine, and then when that happens, you can get sort of a disruption in the tree and get a false impression that this sort of came up ancestrally between the two, when in fact it's got a lineage that's different in the two parts of its genome. So what I'm mostly going to be talking about today is this paper. And I was very fortunate to work with Song Gao, an old friend and collaborator, and my colleague Elena Georgi in our lab, and we also worked with Xiaojun Li, and we tried to sort out what the history of these viruses were thinking about recombination. So it was known that this sequence here, RATG13, was the closest sequence to our current outbreak strain. So we always have 4 current outbreak strains, SARS-CoV-2 in red in these trees. And they're almost identical, which is why you have no branch length out to them. And you can see this bat sequence here among the Cervical viruses is closer to this than any other sequence. So this is This was isolated though in 2013, so this is the kind of evolution you might expect over some years. And it was related by 96.2% sequence identity, whereas SARS-CoV-2 and the first SARS have about a 20% difference. And here's the first SARS epidemic. So we knew that there was a new coronavirus and there was a bat reservoir sequence that somewhat resembled it. And this was published in Zhou et al. In Nature. If you do something called a simplot analysis, you can now compare all sequences and see how close they are to these strains. And you can see that here's our RATG13 that's closest. You can move all along the genome. Here's all 30,000 nucleotides. You can say, okay, out of all of these representative sequences, which one is it closest to? And it's generally closest to the RATG13 throughout the whole genome, but there are these little tiny short stretches where it's closer to something else. And this dark green bit here is a pangolin bit. So remember when I broke apart the trees and looked at the trees in different ways, if you do that, in this case, you can see that there's two regions where it's not closest to this particular bat sequence, region 1 and region 3. So if you look at the trees from those regions, you can see that, in fact, the sequence of the new SARS-CoV-2 sequences are closest to now the pangolin sequence in this little fragment. And in region 1, they're closest to these other bat sequences. So there seems to be a difference in the organization of the tree and the difference in the number of mutations away from these progenitor sequences. Another way to look at this is with something called RIP. It's a tool that we developed that in the early 90s to try to look at recombination in HIV. And you can see this little blue bit is really standing out, and it's again the pangolin sequence. So it's like the RATG13 sequence all the way through the whole genome until you get to this little bit. And this little bit isn't that little. This is 30,000 bases. So it's several. We've got hundreds of bases to look at here. And when we do that, again, we can see this association with the pangolin sequence coming up in this one particular region. And so it's usually the bat sequence, but in this little bit, it's really the pangolin sequence. And this tree is also really interesting to us just when we were looking at all of these many bat sequences in the original SARS sequences, because what we were seeing here was not only is this recombination going on, but in these regions, you can really see evidence for a lot of recombination. Oops, I'm sorry. So this, for example, these sequences, which are usually very distant, can come in to be in a very different place in the tree. So there's a lot of recombination going on in the bat sequences. Here's another case where this one is now coming into this little group of highly related sequences in this region. So I guess the basic point here is that recombination is an extremely important part of the evolution in bats in the reservoir, and these regions are recombining all the time. So when you make a phylogenetic tree of the whole genome, you're going to get an approximate relationship, but in any particular region, that relationship might be violated if you don't try to look at it very carefully. So as it turns out, that region where we have pangolin very close to our current SARS-CoV-2 epidemic is in a critical region. It's the ACE2 receptor contact residue. So this ACE2 is how the virus gets into our cells. And it turns out that it's really, this is now the Wuhan reference strain and a few other of the circulating forms that I've been showing, the 4 forms. And you can see that the RATG13, that's most like the virus, most of the genome, in this really critical region that allows the virus to enter our cells is really like the pangolin sequence. So if it's a dash, it's a perfect match. This pangolin sequence in this region wasn't complete, so we can't look at this one in that region. But this one, it's really a perfect match in all these critical residues. And this is actually quite a variable region among the other bat sequences. You can see the differences. So this is probably a very enabling recombination event in terms of being able to move into the human population. Another thing of interest is that here we go again with our CoV-2, and here's the original SARS epidemic sequences right here in the related bat sequence. And this is the ACE2 receptor binding region. There's a lot of amino acid changes between the first SARS and the second SARS. And despite that, the conformations are really, really very similar. So when you look at it structurally, it can still interact with ACE2 in both of these forms. So that's kind of interesting. These big bright green bits are deletions, and they're in bat sequences. And these sequences don't infect human cells. And so this whole set probably wouldn't be able to get into human cells unless it's somehow recombined with something with a capable ACE2 binding domain. So that was one of the things we discovered when we started looking at these viruses. But another thing that was really striking to us were these regions where you had extensive synonymous substitution. So that meant base changes that don't change an amino acid were just quite high levels. So these viruses were very distant in terms of time in their phylogenetic relationship. But we saw whole regions that had almost no non-synonymous substitutions within this group A of related viruses. So one way you can see that is over here on the right. If a mutation is synonymous, we mark it in blue relative to the pandemic reference strain. And if it's non-synonymous and it changes an amino acid, we mark it in red, and this is in the spike gene. And you can see that you're getting whole regions where there's almost no non-synonymous change at all, because there's almost no red bits. And another way to look at this here is this is the accumulation across this genetic region of synonymous change, which is very level across the whole thing, so it accumulates at a constant pace. But it flattens out, so there's this whole region here, which is actually after the cleavage site and spike, where there's almost no non-synonymous change. So it's really got functional constraints. The protein is not variable in these regions. Only the genetic DNA sequence is changing. So we mapped all of the regions where this is happening in the proteome, where we could see regions where you had almost no non-synonymous change. So the red lines are at the bottom, but you see just a steady synonymous change. You can look at this two different ways. If you look at it with a phylogenetic tree that you base on nucleic acids, you can see there's quite a lot of structure and genetic distance between all these sequences. At the amino acid level, there's almost no change in this particular region. So we just mapped out all the regions where this is happening. So these regions are under very strong functional constraints, and the protein can't alter, although these sequences are quite evolved from one another in all of these particular regions. I just wanted to show one other thing. This recombination, the importance of recombination isn't only in the SARS-CoV-2, but in the original SARS as well. So here's an example, again, of a sim plot, but now this is against the reference strain for SARS-CoV-1. And you can again see that there are these bat sequences, which are mostly quite similar to our virus, but you do get regions of recombination showing up. And there's just extensive recombination throughout these trees. So here's now the first SARS epidemic sequences. These blue are 4 examples from that epidemic. And you can see relationships are very different with these different bat sequences in the different parts of the genome. So there's a lot of recombination going on. It's very important part of being able to get functional units into these viruses. And in conclusion, recombination in cervical viruses is extremely common, and it's a critical evolutionary mechanism, I think, for allowing the jump between species, between host species. And it's quite likely that the acquisition of a complete functional receptor binding motif by an RATG13-like bat-like virus with a recombination event with this pangolin-like virus enabled the efficient use of the receptor and enabled human infection. But I guess I want to make one other point for you guys who aren't used to thinking about phylogenies. These sequences are still quite distant from the actual sequence we have. We don't have a really close sequence showing exactly where this pandemic strain came from. So we have these sort of distant past relationships, and these are the closest things that we've sampled to date to enable us to reconstruct the history. So this pangolin-like sequence might well have come from a pangolin, or it might have come from something else. Same for this bat sequence. This is the closest relative we have, but it's not actually the ancestral sequence. Finally, there's something I didn't talk to you about in this talk. It's discussed in our paper a little bit. The SARS-CoV-2 has a unique Purine cleavage site insertion that's not found in the first SARS outbreak, and it's not really found in other Cervical virus sequences. So it's this unusual amino acid for amino acid insertion, and it causes much more efficient cleavage. So we have both this good receptor binding site and this good cleavage site in our current strain. And these viruses do have insertions and deletions. So we don't know if this came from another bat or if it just came up. So anyway, my acknowledgements are just really the paper again. And the 4 people I have in blue, we worked really closely, very closely together on this, and all the other people in the paper obviously helped in different ways. And I also want to acknowledge again Gizeh, because they just do a tremendous job of bringing these sequences to everybody. And that's my talk. So we can move into pandemic evolution, I believe, for the next talk.
Please go ahead, Joe.
Yes, I will follow this up. Thank you, Betty, by talking about some of the peculiarities of SARS-CoV-2 phylogenetics. Basic points that I'm going to cover today are, one, that SARS-CoV-2 epidemiology and evolution can be better understood through phylogenetic analysis, which if you're attending this, you already believe. But it does require a nuanced interpretation of the pandemic phylogeny, because SARS-CoV-2 doesn't really look like phylogenies that we're used to looking at, like HIV, or if you look deeper in the coronaviruses. So currently on GISAID, there are 67,000 complete genomes reported, and they've been collected since December 24th, 2019. Now, the SARS-CoV-2 genome is about 29.9 kilobases in length, and what I'd say one of its most salient features from an evolutionary perspective is that it encodes a genetic replication repair enzyme, which means unlike other shorter RNA viruses, SARS-CoV-2, like the other SARS-CoV-1 and MERS coronavirus, have a much slower substitution rate. SARS-CoV-2 is about 8 times 10 to the minus 4 substitutions per site per year. So that means, on average, by December 2020, we should expect that newly sampled viruses will differ from the inferred root by only about 24 nucleotide substitutions across that nearly 30,000 nucleotide genome. Now, when you sample a young pandemic as intensely as we have in this global effort, you sample the same genome a lot. And by a rough count, at least 15% of those genomes in GISAID are identical to one another, and that even includes sort of which regions were sequenced. So we have a lot of identical genomes, and most of the highly divergent genomes, those long tips on this mutation tree that I'm showing you here, are likely the result of sequencing errors. There probably aren't actually genomes that are already 25 mutations from the root. So we have to acknowledge that near the tips, things can sometimes get a little funky. And that matters when we want to estimate a molecular clock. So normally when we look at sort of viruses evolving over time, we can think about the amount of time since the root. Here I'm showing you the Washington outbreak clade, and you can see that we have some sequences that were sampled in mid-February, this WAS2, and then other sequences sampled, say, in April. Example I give here is UW5371. Now, the reason I highlight these two sequences, that although they're separated by many, many weeks on this time tree, if you look at the tree and just count the number of mutations, you'll see that these sequences are actually identical. And not only that, but there are scores, actually hundreds of identical viral genomes to WAS2 that were sampled in the Washington outbreak and elsewhere. And you can see those by these dots on the very base of the tree here. Which means that over a period of 44 days in Washington state, this viral genome did not change at all, at least in one particular transmission chain. Now, in other lineages across this tree, of course, mutations did accumulate over February, March, and the beginning of April, but the virus in some lineages actually evolves very, very, very slow. Now, another important point that I'd like to show you on this tree, when we look at it in terms of mutations and mutations that define groups on the trees, we'll call them clades. There are dozens of lineages that all descend from the very same point on the tree. And that means that all of these sort of lineages that are branching off here that sort of form this rake-like pattern that all trace back to WAS2-like genomes, we have no way of distinguishing based on the genetic information alone in which order these transmissions occurred. And we call this a polytomy that comes out of, in this case, the base of the tree. So I'm just going to call it a basal polytomy, which is, I'm trying to avoid a lot of jargon in this talk, but that's one that I just want to get out there so people understand some of the results I'll present later. Now, if we shift to China, where the pandemic originated, and we build a tree of, in this case, about 600 genomes sampled in China during the original stage of the pandemic, so excluding all of the travel-borne cases, you can see that most of this tree is just sequences that their genomes that differ by a single mutation, and that's shown in dark red. So every branch on this tree that is only defined by one mutation is in dark red. Everything that's by two mutations is slightly lighter red, and then 3 or 4 mutations, we get up into the yellows and oranges. Now, what's notable here is that the structure of this tree is very well defined, in that we can really just look at a genome and classify where it should go based on the number of mutations. So you don't need, in this case, a very sophisticated sort of phylogenetic inference to say, well, this group is over here and that group is over here, because they're all defined by the same mutation. Anytime where you have lots of mutations, 3 or really 4 mutations, that's at the tip. And as we mentioned, that's either due to lack of sampling, or also there's possibly some sequencing error going on for the very, very long branches. Now, there is one real 3-mutation branch in the China phylogeny, and that's shown here, where along a single branch, we went from GGG, which codes for glycine, to AAC, which is encodes for asparagine. Now, that's an entire codon shift on a single branch, and we have no evidence of any virus that had an intermediate state. So in this case, even though we have a replication repair enzyme, the virus replaced an entire codon all at once. But this is sort of a very rare event, at least so far in the pandemic, and that most of these lineages are defined by a single mutation. Now, for those of you who are practitioners of phylogeny, I should just caution that when you only have a single mutation on a branch, we can't think about support for that branch in a typical way. And the reason for this is that if you only have one mutation out of 30,000 nucleotides, you can't bootstrap your way into a phylogenetic support. Bootstrapping is where you resample the viral genome with replacement and then reinfer the tree. And if you do that on a branch with a single mutation, you're going to get your expected support levels are essentially terrible, and no one would believe you anyway. The same is true for this ALRT measure of support, which is a measurement of zero branch length. So we can't use traditional likelihood approaches or non-parametric approaches to estimating branch support. Essentially, we're left with just counting substitutions and saying this is a clade. Okay, so that's a bit about what's odd about SARS-CoV-2. Now, how can we put this information into use? And for that, I'd like to present a paper that we have in preprint entitled The Emergence of SARS-CoV-2 in Europe and the United States. This was done with my former doctoral supervisor many years ago, Mike Warby, but also a couple of attendees on this call, Jonathan Pekar and Jeff Joy. My apologies if other co-authors have joined since I last looked at the attendee list. So this is very much a group effort, but I'll be presenting it today. So a brief reminder about COVID-19 emergence in the US. So the first case of COVID-19 in the US was a traveler whose genome was dubbed WA1, and this traveler returned from Wuhan to Washington State on January 15th, 2020. The traveler fell ill and responded to CDC messaging to get tested, which they did, and then they were subsequently isolated. And after extensive contact tracing, no positive contacts were identified, and it was believed that the sort of spread of the virus had been stopped in the US. Then, on February 29th, 2020, a second COVID-19 case in Washington State was identified. And what was remarkable is that this virus differed by WA1 by only 2 mutations. And the inference was that there had been cryptic community transmission in Washington State. And this, as you may recall, led to the lockdowns, which started here in the state of California, and then Washington, and then eventually made its way to New York. And I think it's no stretch to say that the similarity of these cases in the cryptic transmission that was inferred played no small part in that. But what's notable from a phylogenetic perspective was that this outbreak cluster was the only descendant lineage of WA1 in Washington State. So the question we wanted to go and ask was the Washington outbreak a direct descendant of the first known case of WA1 in the United States? And shouldn't we have expected to see other lineages that also descended from WA1 in Washington State rather than just a single large outbreak separated by 2 mutations? So to address this, we, and by we I mean my graduate student, Jonathan Pekar, simulated introduced epidemics of SARS-CoV-2 into Washington State starting on February 15. So we built a scale-free contact network, used an SIR model to model transmission across that contact network. Constructed were essentially timed phylogenetic trees, which we then evolved SARS-CoV-2-like genomes across, and then inferred the tree that you would get from those sequences, and then asked, does this simulated epidemic match the characteristics of our real epidemic? So the characteristics of our real epidemic, if WA1 were the founding strain on January 15th, is that you have WA1, and the virus transmits cryptically, experiences 2 mutations without giving rise to other transmission chains, and then eventually explodes into this large Washington outbreak clade. And we did not see this pattern in any of our simulations. In 70% of the simulations, we also would have found viruses that were still identical to WA1 even a month and a half later. And you remember that's no surprise because viruses identical to the base of this outbreak clade were sampled over a course of 6 weeks. In 95.5% of our epidemic simulations, we observed viruses that were a single mutation from WA1, something not seen at all in the true data. And then in 99.7% of simulations, we'd expect a basal polytomy, that is a lineage branching off from WA1 around the same time as the outbreak clade, but a separate lineage. And then in every single simulation we did, 1,000 out of 1,000, we saw a sibling lineage to the outbreak clade. So again, based on our epidemic simulations, it's unlikely that WA1 could have founded this outbreak clade without leaving other viral evolutionary lineages in its wake. And that was confirmed when we performed a sophisticated phylogeographic analysis taking into account flight patterns and unsampled viruses from China, where you would infer that there would be several Chinese viruses that would occur on the viral phylogenetic tree in between the ancestor of WA1 and the Washington outbreak clade, shown here in red and in green, suggesting a later introduction of this Washington outbreak clade sometime around February 1st. Now, if we turn our focus to Europe, we see a similar story where the first case of COVID-19 in Europe was a traveler to Bavaria, Germany from Shanghai on January 20th. The virus transmitted to a German man whose virus genome is deemed BAVPAT1, which then resulted in a local cluster of 16 individuals. The public health efforts there involved rapid testing, quarantine, and isolation of all contacts, and it is believed that they stanched that epidemic. Until in mid-February, we see the emergence of COVID-19 in Italy, and this virus differs by BAVPAT1 by only a single mutation. And this lineage that then emerged in Italy represents the sole descendant of the BAVPAT1 in Europe. So again, we ask the same question. Did BAVPAT1 give rise to this Italian outbreak, or essentially the European outbreak? And again, just to jump in the beginning, in very few of our simulations, only 6 out of 1,000, did we see a BAVPAT1 leading to only the Italian outbreak and leaving no other descendants or identical mutations. And if we go down to part D, we see that in 99.4% of our simulations, BAVPAT1 could lead to the Italian outbreak, but it would have also had to give rise to another lineage circulating in Europe that would have been sampled. And since we failed to observe that sort of what we call the polytomy lineage circulating in Europe, it seems that the Italian outbreak clade was actually a separate introduction into Europe. And our travel-informed phylogenetic analysis would suggest the same, where BAVPAT1 is shown in this sort of greenish yellow on the bottom, but we see a separate red indicating Chinese strains that would lead to the introduction into Europe, which then subsequently led to the introduction into New York City and New York State and the rest of the United States. So what we're seeing here is a pattern of repeated introduction where the virus made it multiple times into Europe, and actually nearly identical versions of the virus get into Europe and the United States, but don't necessarily take hold, and that early efforts at contact tracing, testing, and isolation seem to have been very effective at preventing further spread of the virus. And that it was only when there were sort of these undetected introductions that that the virus was able to spread and give rise to furthering the pandemic. Now, why do we need more viral genomes, even though the virus is already here? Well, we can sequence now so we can understand later. And a great example of this is in the Beijing market. Now, China, there are, like I said, hundreds of genomes from China, and then, around, well, the first quarter of this year, the number of cases there essentially dropped to 0. And then in June, we have an outbreak in this Beijing market. And we can use the phylogeny to see that the sequences from the Beijing market outbreak, shown here in blue, aren't closely related to any other sequences in China. They're actually more closely related to sequences from Europe and Denmark and the Czech Republic. And this tells us that no, there was not sort of low-level transmission in Beijing over the last couple months, but in fact we have a reintroduced strain that gave rise to this market outbreak. So in that way, the more sequences we can get now from the United States and elsewhere in the world, we'll be able to recognize reintroductions of the virus later. The other thing we could use these viral genomes for is outbreak investigation. Now, as they mentioned in my introduction, I used to spend a lot of time on HIV and understanding whether the virus and sexual partners, and using the virus to determine whether sexual partners were most likely to be potential transmission partners. Now, for HIV, where the virus is a whole lot more diverse, you have a lot of uncertainty about sort of whether individuals are very, very closely related or genetically unrelated, but in between. For SARS-CoV-2, we can essentially ask if you're in the middle of an outbreak investigation, is this virus identical or does it differ by one mutation, in which case they're likely part of the same outbreak. And if there are several mutations different, especially if those mutations are phylogenetically informative, we can determine that no, they are in fact unrelated. And although these people may have had contact and may have been in the same setting during an outbreak, they are not likely part of the same genetic cluster. And with that, I just want to acknowledge funding sources for this, NIAID and CDC, who currently fund me, and Gilead, who is a former funder, but not currently involved in any of my research. So with that, I think we can move on to questions. Thank you.
Thank you very much for these two excellent presentations. And again, I remind everyone to please do put your questions in the Q&A, and we'll try to tackle as many of them as possible. James and Joe, as well as myself, will try to select the questions for conversation. I have a first question, which is, and maybe this is to Joe first, is there value to sampling? It sounds like it's quite valuable to sample viruses from new outbreaks, like, for example, what happened in Beijing, in the late Beijing outbreak, and get insights from doing that. Is there value to ongoing sampling as there's transmission going on within a specific population? And does it matter where we do this? Like, would it be interesting, for example, to sample the viruses that are now being transmitted in South Africa, for example?
Yes, I would say it's valuable to sort of sample just to get a baseline of what's going on in any country or region. But then it's also important to understand, like, if you're doing an outbreak investigation, because that will let you know, depending on sort of the uncertainty of those contacts, you can determine, yes, all of these individuals have identical viral genomes, or all of them differ by one, instead of, well, you know, some of these people gotten infected as part of this outbreak, but you have, you know, some of them who have genetically distinct viruses, and that helps you sort of understand the true scope of an outbreak, now that the virus is circulating on the community level and people can have multiple sources of exposure.
And maybe one last question from me, and then hand it over to James and Joe, is there any evidence that there's an association of these different kinds of viruses that are being isolated and severity of disease? Because that's for, like, for example, mild asymptomatic infection versus severe infections, or we don't know enough about this as of yet?
I would say we haven't seen any evidence of correlation between disease or disease severity and the sort of underlying viral genetic diversity. Now, it's important to note that viral genetic diversity in this virus is very, very young, and I'm sure Betty can talk a bit about whether or not there's a relationship between transmissibility and this virus.
Yeah, I can throw in a comment about that. So we've been following a clade, a group of 4 mutations that come from a common ancestor usually, and it's really moved throughout the globe. And I felt it was likely to be more transmissible because of a very repeated pattern. Whenever this clade entered a community very quickly, even if it entered an ongoing, very intense epidemic with the original form, it would take over that epidemic, and you could track that in region after region. So it became interesting to me as a possibility for increased transmission, and so it had a spike mutation, and spike is really an important protein for two reasons. One is because it allows entry into our cells, so a spike mutation might impact transmissibility or pathogenicity. The other is because it's really the critical protein for vaccines and for immunotherapies based on antibodies. So any mutation in spike, we have to keep an eye on if it becomes common in any region. That's another reason to sequence, actually. If we start seeing a spike variant, maybe a founder virus leads to a spike variant that becomes common in a region where we want to go in with an immunotherapy or vaccine, we really ought to test that variant to make sure that our therapy or vaccine is going to work against it. So those are what was motivating me to look at this particular mutation in spike at 614. And indeed, we found that it is more infectious in pseudotype viruses, and we found that it was associated with higher viral loads in the upper respiratory tract. And those associations are very clear. So this single mutation has these strong statistical phenotypic correlates, but we didn't find any increased association with hospitalization rates in that gene. So it seemed to us that the best evidence we had was, yes, it may be, there's more virus and in a person that could be getting a more a transmissible form, but that didn't seem to be leading to increased hospitalization. However, that same site was studied by another group at NYU in a different way. They looked at fatalities in countries as this particular clade was beginning to grow in frequency, and they found a very strong correlation with fatalities, which was deaths per number of cases that were confirmed, and the frequency of this clade. So there's a report on that's out as well, and I'm blanking on the author's name, I'm sorry, but they're from NYU. Anyway, so they had suggested this in the literature, and there is a paper about that. We didn't see any evidence, and others have not seen any evidence of increased hospitalization so far. So if it's there, it's going to be really subtle in terms of increased hospitalization. Maybe with extreme disease, there is a correlation, and that's what they had suggested.
Thank you both for what were, I found, really excellent and informative presentations. And I think one of the questions that both we as the organizers had, and we saw a little bit in the Q&A itself, was given that this is a global effort, a global sequencing effort that, you know, we have labs across the world really contributing, you know, viral sequences to publicly available databases. And one of the things that Joel brought up was the possibility that sequencing errors might be actually complicating the phylogenetic analysis. We're wondering if there are any sort of best practices or sort of an established sort of criteria to really for you know how people should go about RNA sequencing this virus, given the huge diversity of next generation sequencing platforms we have and the huge variations in methodology.
Can I go first there? I think we're in this remarkable new phase of biology where we're able to look at this virus this intensely, and the global effort's been extraordinary. And this vast number of sequences coming in immediately through GISAID and flowing out to people who are doing the phylogenetics and watching the movement of the epidemic, and people like me who are trying to look for important changes for vaccines. I think this effort's hugely valuable, but yes, there can definitely be problems. And one of the things we do, we have a website called cov.lanl.gov, where you can do the kind of tracking we've been doing. We update it daily, but we have to filter out a lot of viruses. And so we and others do the same thing. We filter out viruses that seem just to have too many base changes, like Joe was talking about. If you get too many base changes, it's sort of not plausible. If there's a whole lot of ambiguities, if there are just funny things in the sequences, we filter out actually tens of thousands of those to get really good sets that we can align. And I think many people who look at these viruses kind of put on those filters. But Nick Goldman's group has got a very nice preprint out on oddities in the database, things to look for. And I think we have to know that there is going to be some error. I think recombination is definitely happening in the pandemic strains. We can see it when we look. It's not a question. Goldman also found cases of it. Those confound phylogenies. So there's things like that happening. So I think what we need to do is realize that this is kind of a gestalt view, and if we have and there's maybe going to be some errors, but you try to filter those out. You can use approaches like Goldman where you look for anomalies and you get rid of all of the anomalies, and I would recommend that paper if you want to try to look into that. So I think there's an intense effort that's growing to try to figure out how to do that as we're absorbing these sequences. But there's also a lot of issues with things like contamination and recombination in the lab bench. And people in different fields have a lot of experience with that, and HIV biologists do. And I just was talking with someone last week who was sequencing from 2 very distant locations and had an identical and rare sequence coming up, and she realized it was a lab contamination that never went into GISAID. So I think education about possible problems with sequences is a really good thing to do so people can mind it in their own labs better. And I believe all of that's happening as we go. We're learning as we go.
Joel, do you have anything to add?
No, I think that covered it.
So I have a question that's sort of combining a few from the chat, and it's basically what can we infer from these phylogenies, both about timing of introduction of viruses to the population and about epidemic size within a population, given that at some points in the epidemic, there have been real questions about the penetration of testing in communities.
Yeah, so with regard to introduction, one of the most obvious things we're seeing is sort of this, repeated introduction of a paper of the Oxford group recently suggesting that there were possibly 1,000 introductions into the UK. And in terms of understanding sort of epidemic viral size, I see one asking about sort of estimating effective viral population size through these models. And it's very important there to sort of correct for sampling biases and to understand sort of that if outbreaks are deeply sampled, which they tend to be, how that can confound those analyses. But I know that there are many groups that are currently trying to work through that, especially out of Imperial College.
Betty, did you have anything that you want to add to that question?
I guess I have a question for Joel rather than wanting to add to it, but I'm wondering, when you were looking in Washington and they had such limited testing, do you have any sense of, in early March, how large the epidemic had gotten? That's, I know, a place you've looked at very intensely related to this question. Could you get an estimate of how many people had been infected?
We were just our simulations that were just based on what the Washington State folks had estimated. I think our final epidemic size for the outbreak clade was about 8,000 individuals on average, and that would have been by March 16th or March 15th. But of course, we were only trying to capture the main epidemic, and there had been sort of other introductions and transmission going on. And as you mentioned with the D614G mutation, that actually was outside of the epidemic clade. Eventually, that epidemic clade in Washington essentially disappears. And as you point out now, everything there is sort of that other lineage. So we weren't looking at sort of the entirety of the Washington outbreak, just this sort of initial founding virus.
Thank you.
So one last question that, and we realize that we're at the end of the hour here, but for both Bette and for Joel, one of the things that we have seen in other viral outbreaks, in particular HIV, is the use of phylodynamics and phylogenetics to sort of guide in almost near real time sort of outbreak analysis for public health intervention. British Columbia has a very famous program that does this, headed up by Art Poon's group. And we were wondering, basically, does that sort of, is there a possible application here to using phylogenetics and phylodynamics in near real time to guide public health interventions? When we saw that Beijing data, that seemed like something that could actually be actionable data if you could trace it back to a sort of foreign strain being reintroduced.
I think that's
sorry, Bette, please.
Ballpark, yeah.
Yes, I agree. I think as the utility of these approaches, sort of where we are right now, at least in certain parts of the U.S., like California, it can help with certain outbreak investigations, but it's not going to help you manage getting the entire sort of pandemic under control here, because we're just too overwhelmed. But we can prepare for the day when things are better and cases are more manageable and investigations can be far more focused and targeted to sort of prevent resurgence. But in the middle of sort of in the middle of mitigation efforts, I think we can just collect sequences that will be useful later.
Fantastic, thank you. And I believe that we're at the end of the scheduled hour. So I know Wafaa had to go. Joe, do you have any last remarks before we close out? I know we didn't get to all the questions in the chat. I apologize for that.
I'd just like to thank Joel and Bette and ICAP for putting this together, and Wafaa, and you, James. Thanks, everyone.
Thank you. I guess that's the end. Thank you so much for all the attendees also for attending this. And we will distribute, I think, a lot of the questions that were asked and some of the, I know that there's been a request for some of the preprints that had been asked for. So we will try to get that out to people.
And the slides and video will be available on the ICAP website shortly.
Thank you.